Bismillahirrahmanirrahim. Assalamu alaikum guys. Welcome to my channel. Today my topic is mechanism of enzyme action. In this topic we will learn how enzyme works and how it has a specificity for a reaction and for a type of substrate. There is a small portion in enzyme molecule that is actually involved in catalysis. It is in the form of depression or pocket on surface of enzyme molecule. This catalytic region is called active site. Mean in on surface of enzyme, mean on top of enzyme, there is a small portion which is like a pockets. Mean it has a depressions up and down. So these depressions are called active site, and the reaction of enzyme is because of this catalytic region. Means this catalytic region is actually involved in a reaction. When an enzyme attached with a substrate, a temporary enzyme substrate complex is formed. The active site catalyzes the reaction and substrate is converted to product. The ES complex breaks and the enzyme and the product are released. The given equation shows these relations, that when enzymes combines with a substrate, enzyme substrate complex is formed. An enzyme catalyzes the reaction and convert the substrate into product, and product and enzyme is released. Now two models have been proposed to explain the mechanism of enzyme action. There are two models which explain the mechanism of enzyme action. These are the lock and key model and induced fit model. Lock and key model. This model was proposed by a German chemist, Emil Fischer, in 1894. According to this model, the active site of enzyme and the substrate have specific geometric shapes. The substrate molecules exactly fit in the active site of enzyme, just like a lock and key. Enzyme is the lock and substrate is the key. This model suggests that the active site is a rigid and non-flexible structure. This model is proposed by Emil Fischer in 1894. The statement of this model is like that active site has a proper and a fixed geometric shape. Mean when reaction is doing, it doesn't change. When enzyme doesn't change its active site shape, it is rigid. Enzyme fits in substrate just like a lock and key. Enzyme is a lock and substrate is a key. Active site of enzyme has a rigid structure and it is non-flexible. During reaction, it doesn't change a reaction. That's why it is specific for a type of substrate. Now, induced fit model. Recent studies do not support lock and key model. On the basis of new evidences in 1958, an American biologist, Daniel Koshland, proposed induced fit model. According to this model, when a substrate molecule fits in the active site of enzyme, the enzyme molecules alter the shape of active site slightly, so that it fits more tightly around the substrate molecule. This model is proposed by Daniel Koshland in 1958. The recent studies means the new studies do not study the lock and key model. So when new studies discovered and according to new evidences, the American biologist Daniel Koshland in 1958 presented a new model which is named by induced fit model. The statement of this model is that when a substrate molecules fit in active site of enzyme, the enzyme molecules alter the shape of active site slightly. Means the statement of induced fit model is opposite to the lock and key model. Its statement is like that active site has not rigid shape, not rigid structure. It has a flexible shape and flexible structure. Because during reaction, enzyme change active site shape slightly. For which purpose? Just to fit the substrate tightly around the enzyme. That's why enzyme fits in substrate fits tightly in the enzyme, and enzyme convert the substrate into product. This is the end of this topic. I hope that you all understand my topic, and please subscribe my channel and like and share my video.